Everyone, we have a guest speaker this morning. First thing, she's here to talk to you about clinical laboratory science.
Hey. Thank you, Dr. Kramer, for inviting us to speak to you. I have a little script because I could talk to you all day about this, so it's not that I won't be able to remember. I need to stay on task. So I'm here to tell you about an under-recognized profession that plays a major role in the health of every individual. I'll assume that most of you are here because you like science. You want to do something in science or healthcare. Maybe you're considering becoming a doctor, a PA, a nurse, maybe a pharmacist. But I'm here to tell you that there are some other options as well. And one particular one that maybe you've never heard of and never thought anything about. So I've worn this white coat for the majority of my career. I'm not a doctor, I'm not a PA, I'm not a nurse or a pharmacist. I worn this coat and I've had the opportunity to explore very different and interesting sides of healthcare. I've been able to do this because of my undergraduate degree. If you found that you enjoy courses like microbiology, anatomy, physiology, genetics, or biochemistry, something called clinical laboratory science might be a profession that you'd be interested in, especially if you're questioning whether or not you want to work directly with patients. It may also be the start of a pathway that leads you to a rewarding career that may be a little bit different. So I'm here to tell you that UNC offers both a bachelor's degree and a master's degree in clinical laboratory science. Our undergraduate degree focuses on educating and training students to perform clinical laboratory testing in a variety of areas. Those areas might be clinical microbiology, hematology, blood banking, or what's usually called transfusion medicine, clinical chemistry, a variety of areas in the medical sciences. Our students rotate through laboratories at UNC hospitals, at Duke Medical Center, and the VA hospital in Durham, and work side by side with other clinical laboratory professionals performing tests that are very important to patients that help make diagnoses. We also offer a degree, a master's degree, in clinical laboratory science with an emphasis in molecular diagnostic science. Those students train to perform tests in areas of oncology to help diagnose cancer, infectious diseases like HIV, hepatitis, and a variety of other viral pathogens and things like that. They also test for inherited disorders like cystic fibrosis and a variety of things that affect every individual from the time of birth until they die. So our graduates have gone on to do some very rewarding things and have a variety of careers. And as you can see here, not all of our graduates stay in the laboratory. They've done a variety of exciting things, and I think this slide speaks for itself. If you'd like to find out more, please check out our website. That was on the previous slide. After that. We're offering information sessions this fall. There's sit-ins in our lecture courses. There's open labs, so you can see what the student laboratory is like. We also give tours around the hospitals. You can take a look at the clinical laboratory there. And you can also meet face-to-face with faculty. So hopefully pique some interest here in this classroom. And check out our website, sign up for some of the opportunities to explore this meeting. Thanks for your time.
Thank you. I just have two questions for you. What's the average employment after you graduate from clinical laboratory sciences?
For those that want to work, 100%.
100% employment after you have a 2-year degree in clinical laboratory sciences. You can enter into clinical laboratory sciences as of your junior year here and graduate with a 4-year degree. 100% employment. And what's the average salary?
Average salary now, I think, well, for a starting graduate, it's anywhere from, depends on where you go to work, 47 to 50,000 right out of school.
Right out of school.
Yeah.
100% employment.
So the average is about 62,000 a year for those that are work.
I just wanted to point out those numbers. So thank you so much.
There's some flyers here and some little business cards with their website too, if anybody's looking.
So today we are going to continue on with immunology. Immunology is also a subject that they will go back over in clinical lab sciences so that you understand when you're sorting for stem cells, for bone marrow transplants, or doing all sorts of tests that you have to understand what the stem cells are and what your tests are running. But I think it's very interesting. Also, the master's degree program is a good program for people who are already maybe juniors or seniors who are looking to do something. Once you get your master's degree program, it's generally pretty easy to go into something like medical school or even law school, something like that, from a master's degree in clinical lab sciences. So I'm just saying, I'm not pushing it, I'm just saying that they're alternates to nursing, pharmacy, stuff like that. So this is acting really weird today. So we are going to continue on. There'll be a poll everywhere question about middle of the way through. But today I just kind of we're going to finish talking about the cells that we had started talking about last class. And so these are the troops. These are the troops that play a role in defending you or in trying to figure out what to do in order to make sure that if you have an infection, you can fight it off appropriately. So let's talk about these troops. We talked about granulocytes. The only granulocytes that are granulocytes are the ones that we can see granules in their cytoplasm, hence granulocytes. Eosinophils, basophils, and PMNs or neutrophils. Those terms, PMNs or neutrophils, are in your outline as interchangeable terms for those cells. We're going to use them interchangeably in class. So we have these granulocytes. We also have mast cells. And mast cells are also a granulocyte, but notice they're separate from the regular granulocytes. The regular granulocytes are the ones that circulate in the bloodstream. The mast cells are found in the tissues, and generally they're found in the tissues anywhere you have a circulatory system, like every place you have a capillary, every place you have an artery, any place you have a vein, which is where? Everywhere. So mast cells are in your tissues everywhere, and mast cells are kind of one of the alarm cells that when we talked about that paper cut notifying you immediately that you had a paper cut, it was due to one of your mast cells being released and notifying that something had gotten through the wall in your tissues. Okay? So that's why we need mast cells. But we're going to talk about eosinophils, basophils, and neutrophils. But the main one that we're going to talk about are neutrophils. And the reason that we're going to talk about neutrophils is because they are the most important cell. They're one of the most important cells in the innate immune response. So what do I mean by that? So let's go on to this next slide. So this is something that we're going to be talking about. Notice here, when I talk to you about mast cells are lumped in with basophils. At one point, we thought that mast cells and basophils mast cells were derived from basophils, and then we found out that they're two completely different cells. One is tissue, one is circulatory system. But I want you to know that in your tissues, you have a huge number of mast cells, where what's the amount of basophils that are circulating in your blood? Very, very few. They're a rare cell. So we hardly ever talk about basophils that much. We'll talk about basophils when we get to something called allergies. Okay? Or when we talk about helminth infections. The other thing that we're going to talk about eosinophils. Eosinophils, what's the percentage of the total white blood cell count? So you guys have all heard of a total white blood cell count every time you've watched Grey's Anatomy and you do a complete CBC. So that's a complete blood cell count. So they're looking at normal numbers. So when we're looking at normal numbers, if the numbers for these cells are above what we normally see, say for eosinophils, normally we see about 2 to 4% of the total white blood cells are eosinophils. If all of a sudden we saw 15% of your total white blood cells as being eosinophils, we would say that you had eosinophilia, or you'd be have a high eosinophil count, and we would think that you would have something like probably a helminth infection, because eosinophils are primarily responsible for participating in immune responses to parasites. So we would say that you'd have something like a parasitic infection, but because we know that allergies and asthma mimic a parasitic infection, it might be that you're having a hyperallergic response or some sort of an asthma response to something in the environment. So we're going to look at the total numbers of cells that are there. So basophils and eosinophils are primarily responsible for responding to these parasitic infections. They are not phagocytic cells. They degranulate in response to an infectious organism. So they're not going to eat the infectious organism. They're going to throw their toxic granules out. And if you think about what we talked about before in the last class, we talked about how the different granules stained differentially. Some of the granules stained with a neutral dye. Some of the granules stained with a basic dye. Some of the granules stained with an acidic dye. Now, that should tell you that the contents of those granules are inherently different from each other, right? So they have different functions, different reasons to do things. So for eosinophils and for basophils, their job is to have granules that help take out parasitic infections. That's the primary role that they were meant to do. Mast cells are an alarm system. We're going to talk about them in context of inflammation. Now we're going to go on to the big one. We're going to go on to polymorphic mononuclear cells. So PMNs, polymorphic nuclear cells. And I say this every semester. We are not rocket scientists as microbiologists. If you have a polymorphic nucleus and you're a cell, what are we going to call you? A polymorphonuclear cell. So what I mean by this is that here you can see the nucleus on a PMN is this kind of weird, wacky shape. Sometimes there's 3 lobes, sometimes there's 4 lobes, sometimes there's 5 lobes. The nucleus is different in every PMN. It's polymorphic nucleus. PMNs. It's also the cell that's stained with a neutral dye, right? So it's also known as a neutrophil. Just like a car and an automobile are 2 interchangeable terms for the same thing, right? So here we have a polymorphonuclear cell, and look at the number of cells we have for that. 55 to 65% of all your circulating blood cells are going to be PMNs. They are the first line of defense. They're one of the first infiltrating cells when you had your paper cut. When you see pus, the majority of all pus, that white, are dying and dead neutrophils that have tried to eat bacteria. Now, the other thing is we talked about basophils and eosinophils being specific for parasites. PMNs are generally specific for extracellular bacterial infections. It's extracellular because PMNs can't eat something hiding inside of a cell. So if it's an intracellular bacterium, that bacterium is safe and sound from PMNs. They can't get to them. They can't eat them up because they can't see them. So when we talk and when we see elevated PMN levels, so 55 to 65% is normal. You'll need to know that 85%, if I said, all of a sudden in our complete white blood cell count in an exam, I see 85% PMNs, that should immediately tell you, it should immediately make you think, 1, inflammation, 2, bacterial inflammation. Because the job of PMNs is to eat and destroy extracellular bacteria. Gram-positive, gram-negative, it doesn't matter. Okay? So, it is the most common type of circulating leukocyte. But again, let's break down those words in that phrase. Circulating leukocyte. They are found primarily in your bloodstream, primarily in your circulatory system. They only migrate into tissues or into a site of infection when inflammatory signals are sent to say, hey, I need you to get out of the bloodstream and migrate into my arm where the paper cut was. Okay? Unless there are inflammatory sites, information saying that we need you to come to this specific site, unless we get that information, then all that's going to happen are the PMNs are continually circulating in the bloodstream. The other thing that happens are, these are one of the most primary cells, they are the most numerous cell, and they have the shortest half-life of any cell in the body. Meaning they live about 3 days if nothing is happening. So if you're normal and healthy, 3 days, the PMNs that you made 3 days ago have died. If they're fighting off an inflammatory response, it might be that they use up all of their granules, they destroy themselves after maybe a couple of hours because they've been eating up and destroying bacteria so much that they've utilized everything that they can do. And then, one of the things PMNs do are they eat and destroy. We're going to talk about a phagosome, and then the lysosome, that granule is going to bind with in the rest of this class. We're going to talk about this. Once all those granules are used up, the other thing that a PMN can do, it's used up all its granulocytes. It's eaten as many bacteria as it can, but it still sees there's lots of enemy out there. If there's lots of enemy out there, the one last thing that it does is it actually self-destructs and it throws its DNA out into a net, and we call that a neutrophil net. And their DNA explodes onto a...
You're finished what you're saying.
Okay. The DNA explodes onto the bacteria in the area. That DNA is sticky. It binds to the bacteria, and then other PMNs have receptors to bind to that DNA, pull them in, and eat them. And it's like a net, a fishing net. So instead of just eating one bacteria, one bacteria, one bacteria, all of a sudden, I've got a net of all y'all bacteria right here. So it's a neutrophil net.
Okay, so is the nucleus, the weird-shaped nucleus, what allows that to happen?
Is it the weird-shaped nucleus that allows that to happen? I don't know. It's a PMN. They are great at eating and destroying bacteria. They have all sorts of mechanisms that they have. We're going to talk about how they bind to and how they know what to eat. There are two main things. They're phagocytes. They're one of the 3 main phagocytic cells in the body. This is one of the primary phagocytic cells. Is an eosinophil a phagocyte? No. Is a basophil a phagocyte? No. Is a mast cell a phagocyte? No. And there are other websites that I have seen online when people are saying, is an eosinophil really not a phagocyte? It is not a phagocyte. Trust me, it is not a phagocyte. We'll talk about why in a little bit. Okay? So, we have those granulocytes, and those are the granulocytes. These are the only granulocytes that you need to know. Okay? The other thing that PMNs can do is anything that's coated in certain kinds of material, they eat. So if it's coated in antibodies, we're going to talk about antibodies in the next class, they can eat those. Certain kinds of antibodies. So we'll talk about them. And we'll talk about antibodies that allow eosinophils and basophils to work. There are 2 different kinds of antibodies that work for PMNs and eosinophils, and that makes sense because eosinophils and PMNs have different functions. It makes sense that they would have different antibodies that have different functions as well, right? I know. This is all kind of stuff I'm throwing at you, and we're going to keep going over and going over and going over as we go through this part of the class, okay? So the next thing, I want to show you, this is a mast cell. So that red spot, we're staining specifically for mast cells. This is the alveolar, this is the lung of the tissue. So even in your lungs, anything that gets through your lungs is going to activate mast cells. Any place that you have capillaries, there's mast cells outside of that capillary waiting to be activated. They are alarm responders. They release their granules to tell the immune system that inflammation is happening here. And we'll talk about how the animation for inflammation is something that I test on every semester. You need to know all of the animations that we're going to talk about, but the animation for inflammation we're going to go over here when we get to inflammation. It's important. So that is mast cells. That's where mast cells are in the tissue. They're not in the circulatory system, they're in the tissue. The next thing that we're going to talk about are these mononuclear phagocytic cells. And we used to call them other names. We used to call them monocytes, macrophages, and dendritic cells. And they're still listed as that in the book. We still identify them as that in the book. Normally what we find with monocytes are circulating precursor cells. And depending on the kind of fight we find ourselves in, there are clues given, we're going to talk about them as cytokines, at the site of the inflammatory response that's going to drive that monocyte into either becoming a macrophage or a dendritic cell. And we used to believe that once you were a dendritic cell, you were a dendritic cell. Once you became a monocyte or a macrophage, you became a macrophage, and that was all you could be. Now we've been able to track these cells, and we find out that instead of just those two arrows going down, we now have this red arrow going across, and if this cell needs to morph into becoming a dendritic cell, it does. We used to have people have all of these papers, these scientific papers where they've gone online, where they've said, this is a macrophage and this is a dendritic cell. And people say, no, this is a macrophage and this is a dendritic cell. And people say, no, I found this population that's a macrophage and this is a And then we decided that basically they're just all kind of the same cell. And they might be morphing between one and another when we're identifying them in a laboratory. So these are mononuclear phagocytes. Both dendritic cells and macrophages are phagocytic cells, depending on wherever they are. They have different reasons for being a phagocyte. Macrophages are really good at cleaning up debris, cellular debris, and in normal tissue, if one of your cells dies, it's a macrophage that's in your tissue that's kind of like a garbage man that gobbles up that dead cell so it's not just sitting around gunking up the place. Okay? So macrophages eat dead and dying cells, they clean up debris. They also clean up debris during an inflammatory response, but they also are effector cells that we're going to talk about in a minute. Dendritic cells really only have one function. The function of a dendritic cell is to activate the acquired immune response. That's its job. We'll talk about how it does that in a little bit. We'll talk about dendritic cells more in the future. It is actually these two cells is actually dendritic cells. Here you can see a dendritic, I'm going to go back. It's actually these two cells that go on to activate T cells and B cells. Macrophages and dendritic cells are something also known. They're one of the three cells that are also known as an antigen-presenting cell. So, macrophages and dendritic cells are one of the three cells that are phagocytes. Macrophages and dendritic cells are one of the three cells that are antigen-presenting cells. Okay? So now let's talk more about other kinds of macrophages. During your development, when you're born and when you're in utero, you're going to have precursor macrophages that migrate into specific tissue. And once they migrate into specific tissue, they differentiate into different kinds of macrophages. These macrophages never change. Once you're a tissue-specific macrophage, you will always be a tissue-specific macrophage, but your job is also to help with inflammation, help clean up, help structurally support the tissue you're in. So when we talk about these macrophages, I'm going to ask a question about the different names of these macrophages, but generally they're pretty straightforward. So when we talk about macrophages, we're going to talk about alveolar macrophages, and alveolar macrophages make sense that they're going to be the macrophages that are found in the lungs. And you guys know from anatomy that there are microglial cells in the brain. And did you know that microglial cells were macrophages? So they're a type of a macrophage that is specific to brain tissue. And you know that there's a structural component in those microglial cells as well. They play an integral role in the brain. They play an integral role in Alzheimer's disease, in brain inflammatory responses. So we have microglial cells in the brain. We have mesangial phagocytes in the kidneys. Mesangial phagocytes are there because the kidney is there to filter stuff out of the blood. So the stuff that gets filtered out of the blood has to be eaten and destroyed, and so those mesangial phagocytes are there to help clean up the stuff that's being filtered out through in the kidneys. We also have one of the other ones that's not was not listed there, is in the bone. We also have osteoclasts and osteoblasts are macrophages that are found in the bone. So those are precursor cells that have gone into the tissue, have developed into specific cells that never change. They are a specific kind of macrophage. Okay? And they make more macrophages. Kupffer cells make more Kupffer cells. Microglial cells make more microglial cells. The other thing that we're going to talk about, and this has just in this new edition of the textbook. In the previous edition of the textbook, we never used to talk about M1 versus M2 macrophages. What we now know are those mononuclear macrophages. Those mononuclear macrophages, when they're being generated, can be generated into two, and we're probably going to find more, phenotypes. An M1 phenotype is something that instigates inflammation. It is a pro-inflammatory macrophage. It's going to make pro-inflammatory cytokines. It's going to drive PMN responses. It's going to drive an antibacterial response. It's going to drive inflammation. An M1 macrophage. When that monocyte becomes a macrophage, it might become an M1 macrophage. However, it might also become an M2 macrophage depending on the signals that are being sent. And an M2 macrophage is the exact opposite of what an M1 macrophage is. An M2 macrophage helps repair the tissue, helps after the inflammation is over, after the war has gone through, after the bacteria have rampaged in your system, we need to clean up, we need to repair the tissue, and we need to get reset. And that's what an M2 macrophage does. It does tissue repair, it helps angiogenesis, meaning it helps repair the blood circulation, the capillaries that might have gotten hurt during an inflammatory response. What we also know is when these aren't created properly, we can see disease. So what do I mean by this? I mean that for people who have diabetes, type juvenile diabetes, they have a pro-inflammatory environment in their islet cells that make insulin. Something has triggered an inflammatory response in their islets, and what they found in those islets, instead of M2 macrophages to try and shut down the immune response to self, we find M1 macrophages that are driving an inflammatory response that are saying, no, I don't know what those islets are. We need to kill this cell. Something is wrong with those cells. And so we see in people who have autoimmune disease a higher level of M1 macrophages instead of M2 macrophages. And you know it's equally wrong and bad? In cancer cells, that cancer tumor tissue is full of M2 macrophages. M2 macrophages because that cancer cell has programmed the macrophages that have migrated into that tumor to say, I need you to tell them these are not the cells you were looking for. Go away, all is fine. I need to make circulatory systems. I need you to go away. The cells come up to fight the infection. The M2 macrophages secrete cytokines and markers that say, no, you don't. We're fine. And then the cancer grows instead of being attacked. So we are trying to find in current cancer therapy ways of flipping those M2 macrophages back to an M1, a pro-inflammatory phenotype that can drive an anti-tumor response instead of allowing the tumor to grow. So this is always what happens when things go amok. When something happens, something goes wrong. How does the cancer cell tell these macrophages to become M2 macrophages? We don't know yet. The person who discovers that might be one of the people who are getting a Nobel Prize in the future. All the Nobel Prizes are coming out this week. Okay? So these are those cells. Any questions about macrophages? Uh-huh.
Are you going to be responsible to know all the different names of cells on this diagram?
Yes, you are. You're going to be responsible to know the names of all the different cells on this diagram because those are important cells. Because we're going to be talking about them throughout the rest of the class. We're going to be talking about them when we talk about kidney infections, when we talk about liver infections, when we talk about, Kupffer cells are in the liver, but really there aren't that many there. The other cell that you can see that resident and circulating macrophages in the lymph nodes. We're going to talk about lymph nodes in a little bit too. Okay? So those are just regular macrophages that have become macrophages via signals from the cell. But peritoneal macrophages make sense that there are macrophages in the peritoneum, in the space in your gut. Okay? Okay. The other thing that you're going to need to know are that there are specific, just like there are site-specific macrophages, there are site-specific dendritic cells. And these site-specific dendritic cells have specific names. One of the most important specific dendritic cells that we have is this cell called the Langerhans cell. This Langerhans cell is a specific kind of dendritic cell. So not only are the mast cells there to warn of an infection, remember I told you about the wall. And the bad thing about the wall in the books is the wall in the books, they didn't have enough troops to man the entire wall. They were low on troops, so they put up fake soldiers in some place, and some places of the wall just didn't get patrolled. Not so in your body. What we have here is we have the Langerhans cells that is in the epidermis. So it's in that stratified squamous epithelium. And notice that these dendritic cells have these, they were called dendritic cells because they have dendrites that they thought they looked like nerves. But instead of dendritic cells, these are just long cellular processes that are welling and waiting to be triggered to say something has gotten through the wall. And notice that when you look in this, these cells have been colored green. We have stained them with green, with a green fluorescent dye. Notice that there is almost no space that is not protected by some dendritic extension from these dendritic cells. So when your skin is cut, your dendritic cell that is in an immature state in your skin says it has been activated. Once it sees bacteria or once it sees pathogens, it drives those dendritic cells to become mature and then to go on to activate the acquired immune response. PMNs will migrate into the system where the wound was, while the dendritic cells are going off to activate the acquired immune response. So the PMNs and the macrophages that are in tissue will start fighting off their response. The dendritic cells migrate. Also, we have these dermal dendritic cells, and just like those Kupffer cells, or just like those alveolar macrophages, these are tissue-specific dendritic cells that are different from the mononuclear phagocytes that we talk about when we talked about the mononuclear cells. These regenerate in the epidermis. These regenerate in the dermis. And these make sure that you are protected and covered, and that anything that gets through is identified and alarms have been properly sent off. Okay? So, dendritic cells, Langerhans cells, dermal dendritic cells are specific names for a type of dendritic cell. Mononuclear dendritic cells are ones that have developed from a mononuclear cell. Are we good? Okay. Now, the other thing that we're going to talk about is we're going to talk about cytokines and how cells talk to each other. So just like we talked about viruses, viruses can't bind a cell unless their specific surface spike can bind to a specific tissue ligand. Okay? Now, if you're trying to talk from cell to cell, there are ways that we talk from cell to cell. One of the ways that we can talk from cell to cell is some cells have to directly interact with each other in order to talk. So what do I mean by that? That means that I'm going to have, again, I'm going to find another person I haven't picked on. I'm going to find you. I haven't picked on you. So I am a cell that needs to communicate information to you. You have a receptor, upregulator receptor. That's good. You can upregulate both. She has receptors on the surface of her cell. I am a cell that needs to tell her information. I'm going to migrate to her. I'm going to bind to her, and this binding process sends a signal down her receptor into her nucleus that says, you have been selected, you have been activated, and you need to respond. Will she be activated? No, because she has not been bound. She is the one I chose. She is the one who got activated specifically by having interactions. Now the problem is that it depends on if it's a receptor or if it's a ligand. Depends on when it was identified. Was the receptor identified first? That's what we call the receptor. Then we search for the ligand for that receptor. We're going to talk about a set of receptors and ligands. One of the receptor is called CD40, and one of the ligands is called CD40 ligand. Which one was discovered first? CD40 was. Then we found out what CD40 bound to, and we called that the CD40 ligand. They would be reversed if the other one had been found first, but it wasn't. Okay? So now, some cells have to have cell-to-cell contact, and those are specific and regulated. The other thing that I want you to do is anybody whose birthday is in October, I want you to upregulate the birthday receptor. So upregulate your birthday receptor if your birthday's in October. Now I want you to look around, raise your hand high so we can see you. Notice how many people have raised their hands. Is everybody raising their hand? No. So I want you, this is what upregulation is. People didn't have their arms in the air. They have now upregulated a ligand that wasn't there before. Now I want you to downregulate that ligand. Okay? When you downregulate that ligand goes away and you can no longer be activated. So when you upregulate something in response to certain chemical signals called cytokines, or you might be the cells that raise their hand in response to the October ligand are the cells that are supposed to be responding to this particular infection. And everybody else is supposed to mind their own business. If you don't have a ligand that is going to bind to a specific cytokine, you are not the ones we need. And if you start charging in where you're unwanted, you might make things worse. You actually will make things worse. We only want those people who upregulated their ligands. Okay? So, when we talk about that, we're going to talk about specific tissue interactions where it's cell-to-cell contact, receptor ligands, or we're going to talk about cytokines. And cytokines are a way for one cell to send a message to everybody in this room who was October birthdays. So everybody who's got October birthdays, upregulate your October ligand. Everybody upregulate your October ligand. Some people over here have stopped upregulating. Okay? Those are the people that I'm going to send a cytokine out, and because I can send a cytokine out to them, they bind the cytokine, that's going to send a signal, you're going to downregulate that signal, and that now has sent, instead of me having to go out and touch you specifically, the cytokine that I made, bound to you, sent a signal into your nucleus that said, I bound this specific cytokine. Ooh. Creepy. Somebody wants to come in. Ghosts. They love immunology. Okay, so that. What the heck? Okay then. Okay, so we have cytokines, specific kinds of cytokines, and people are like, oh my God. Do we need to know these? You need to know the ones that are on your outline. When I talk about cytokines of the innate immune response, the most important set of cytokines that drive inflammation are three: IL-1, IL-6, and something called TNF-alpha. So when we talk about IL-1, IL-6, and tumor necrosis factor, IL-1, IL-6, and TNF are generally always secreted as pro-inflammatory cytokines. For those of you guys who's like, why do I need to know these and where they come from? The reason that you need to know these and where these come from is because, for example, if you are going to go into pharmacy, not only are you going to have to know these cytokines and their names and what they do, but you're going to have to know every signaling molecule that sends the signal into the nucleus so that you as a pharmacist can interrupt inflammatory responses and make an anti-inflammatory drug that will make you millions, billions. Okay? That's why you need to know these. And the reason, just so you know, this is the smallest list I can make. There are something like, I think now we're upwards of 80 or 90 different cytokines that are important in the whole immune response. There's going to be a whole new list in the next book that people need to know. Right now, you only need to know the ones in your book and in this figure and in your outline. Pro-inflammatory cytokines that are important are IL-1, IL-6, and TNF. We're going to talk about their sources in a minute, okay? As a matter of fact, one of the things we're going to talk about is when we get to inflammation, we'll talk about IL-1. We're going to talk about IL-6, and we're going to talk about TNF. But most of the time, the cytokines that make IL-1, IL-6, and TNF are all driving an innate immune response for inflammation. Those are the ones that happen. IL-4 is something that's going to help, let's see, what are the cytokines that are in your outline? Let's break them down. We're going to talk about interference. We're going to talk about, let me go back. Oh, I skipped over. You guys didn't stop me when I skipped over. I'm going to talk about these cytokines and then we're going to come back. So we're going to talk about interferon alpha and beta. We're going to talk about interferon gamma. And then the cytokines that act specifically on the acquired immune response, we'll talk more in the next lecture. But those cytokines are IL-2, IL-4, IL-10, and IL-17. Okay? Just so you know, we're finding out all of those cytokines that act on the acquired immune response also act in the innate immune response. But we have more information about them in our book for the acquired immune response. So we're breaking them down in this class into those groups. But we'll talk about specifically what they do in just a minute. Okay? The other thing that I want to go back and talk about, we're going to talk about lymphocytes, natural killer cells, innate lymphocytic cells. Let's go back to number 4 under the cells. So one of the things that we talked about when we talked about cells, let's go back to these cells. We didn't talk about NK cells, and we didn't talk about B cells and T cells. The reason that we're not going to talk about NK cells until we get to the acquired immune response is because they're an innate cell. They're a natural killer cell that doesn't need training, but it acts in context with one of the T killer cells. It's an innate cell that has an acquired immune component. So we have an innate killer cell and then we have an acquired killer cell. The same thing is true for ILCs, for innate lymphocytic cells. And again, we just discovered innate lymphocytic cells literally 2 or 3 years ago. We did not know what they were. We did not know where these cytokines came from. We could not identify them 2 years ago, and now they are huge, and they play a huge role in our understanding of how an immune response works. So, innate lymphocytic cells, there's an innate cell that's not a T cell or a B cell, that has a similar component that is a T cell and a B cell in the acquired immune response. So we're not going to talk about the innate lymphocytic cells until we talk about the lymphocytes themselves. And then we talk about which ones are innate and which ones are acquired and how they function. Most of the time they function almost exactly the same way. The only thing that's different is how they're triggered. Okay? So we'll talk about that. Does that make sense? Okay, so let's talk about, we talked about T cells and B cells. We'll talk about those in the acquired immune response. Anything that I've missed or anybody want to ask a question about so far? Okay? Okay, let's move on. So we're going to talk about, right now for the cytokines, the only ones you need to know are the ones that are on this outline. Okay? Next semester, there's going to be more. Now we're going to talk about other ways that the immune response knows something is happening in a cell or in your body. So we have the paper cut, or we breathed in a respiratory virus, or we've had some sort of bacterial infection from for whatever reason, you have a bacterial infection, okay? One of the things that we need in the innate immune response are we need alarms. We need an alarm to go off when something gets through. So now, you rob a bank. The person who robs a bank is a bank robber, correct? Do we know what kind of bank robber they are? Do they know what kind of heist they've pulled before? Do we know their specificity? Do we know anything about their previous thing other than the fact that they've just robbed a bank? No, we don't. So this is what we're talking about with these pattern recognition receptors. Pattern recognition receptors, and we abbreviate those PRRs, and you're going to need to know what a PRR is on the exam because we always abbreviate them PRRs for pattern recognition receptors. I'm going to tell you, you're going to hate abbreviations by the end of this section. You just will. Because what PRRs bind to are either PAMPs or MAMPs. A PAMP is a pathogen associated molecular pattern. So it's a pattern that's recognized by those receptors. So it's either a pathogen-associated molecular pattern or it's a microbial associated molecular pattern. So we have PAMPs and DAMPs, or PAMPs and MAMPs, but we also have DAMPs. And DAMPs are due to damage. So it's damage-associated molecular patterns. So when you get hit by a bat, or you're playing softball and the softball hits you in the face, there's no microbes there, but you're having a damage, and what's released are damage that is recognized by these receptors. It's a pattern of release. So we have PRRs, these pattern recognition receptors that are found on the surface of cells. We have PRRs that are found in endosomes, and we'll talk about why they're in endosomes in a minute. We also have PRRs in the cytoplasm. So what are these PRRs? There's more abbreviations coming. Yay. Wait for it. Okay. There are things like TLRs. Yes, you're like, oh my God, seriously. It's true. They're named TLRs, and they're named TLRs because the people who studied Drosophila named their protein toll proteins. And they identified them as being an inherent, innate immune cell, or a response in flies, in an earliest precursor organism. We found similar receptors in humans, so we called them toll-like receptors. They look like those Drosophila molecules, only they're found in humans. Toll-like receptors are very specific. Toll-like receptors, let's go through what toll-like receptors bind and where they bind. We have toll-like receptors that are on the surface. Toll-like receptors, all toll-like receptors are bound in a plasma membrane of some sort. Either they're on the surface of their cell or they're in endosomal membranes. So let's look at these. We have a toll-like receptor that is on the surface of a cell, and it binds something called peptidoglycan. What is peptidoglycan? It's a bacterial cell wall. It's a pattern that's conserved among all bacteria, and if we see it, there's a frickin' bacteria here. If that bacteria isn't supposed to be here, we need to alarm the immune system that there are bacteria here. It has gotten through our defenses. Peptidoglycan. What's another on the surface of the cell? Something called lipopolysaccharide. Have you heard about that before? Okay, let's talk about another molecule that was just on the exam called flagellin. It's not flagella, it's not flagellum, it's flagellin, and it's the protein that makes the filament of only bacterial flagella. If we find the protein flagellin in your body, that means there's a frickin' bacterial flagella there, and there's a bacterium swimming around my system, and I need to make sure that we fight it off. Alarms go off. We good? Now, when we talk about endosomes, endosomes become acidified. And when they acidified, anything that's been internalized in that endosome is breaking down and destroyed. We're going to talk about these TLRs in the endosome and look at the TLRs in the endosome, what they bind to. Oh, look, they just bind to double-stranded RNA. Double-stranded RNA, when we talked about making RNA from a single-stranded RNA virus, it always has to go through a negative stranded phase. Double-stranded RNA. Any RNA virus is going to induce double-stranded RNA. Most DNA viruses can also make double-stranded RNA at some point in their life cycle. Double-stranded RNA is an indication of viruses present. If we see double-stranded RNA, it's a virus. So can these toll-like receptors differentiate between bacterial infections and viral infections? You betcha it can. Because we're going to have to make a different immune response for a virus than a bacteria. Okay? So, it also detects bacterial DNA, and the way that it detects bacterial DNA is because the methylation pattern on bacterial DNA is different than the methylation pattern on human DNA. We methylate our CGs. Bacteria methylate their A's. If we find DNA that's methylated on the A's, the body says, holy cow, there's something wrong here. This is bacterial DNA. It's a problem. Okay? And then we have single-stranded RNA, and one of the ways that we can also identify bacterial RNA or single-stranded antiviral RNA is if there's no cap or polyadenylation motif on this bacterial RNA. If there's no cap or bacterial motif, then it's not human and it's alarms. Are we good? So some viruses always live in the cytoplasm. So the viruses that live in the cytoplasm, we need cytoplasmic detectors, and we call them NOD-like receptors, NLRs, or RIG-like receptors, RLRs. The NOD-like receptors are generally things that make, as a matter of fact, in your notes, we talk about an inflammasome. It's something that's going to recognize mostly bacterial infection. And when we look at the NLRs, it detects similar things. It detects flagellin, it detects damage, it detects peptidoglycan. At UNC, another person, is there a big NLR missing? We talked about how important one specific gram-negative molecule is. Is there a NOD-like receptor missing in that component? What's missing? LPS? Is it LPS? Somebody say LPS. It's LPS. LPS is missing. You would think there should be a NOD-like receptor for LPS, right? Because it's huge. The person who discovered the NOD-like receptor before this book was printed is Dr. Ed Meow, who works on this campus and might get a Nobel Prize for recognizing and identifying, helping recognize and identify these sometime in the future. Jenny Ting is another researcher who helped identify these when they first came out. Probable Nobel Prize candidate in the future. Big names. And yes, his name is Meow. And he loves cats. Okay? So, we have NLRs that primarily identify, they cause an inflammasome. The book talks about an inflammasome. They cause an inflammasome. They primarily detect bacterial motifs, patterns. We also have our RIG-like receptors, RIG1-like receptors, RIG-I-like receptors. Sorry, it's RIG-I-like receptors. But we abbreviate them RLRs. And those specifically identify viral motifs. And they induce something called interferon alpha and beta that we're going to talk about in just a minute. Okay? We're going to talk about them. You guys are going to have a pull everywhere question in just a minute. So I want you to start getting pull everywhere question because you all are falling asleep on me. It's very warm in here. I know. You guys just ate lunch and it's all going to your gut. Okay? So we're going to talk about inflammasome. We're going to talk about how we stop, what happens when these motifs, and what happens when these motifs bind their ligand, and the ligand for toll-like receptor that binds to LPS is LPS. the ligand that binds to the endosomal TLR is double-stranded RNA. Those send signals into the cell that say this kind of pattern has been recognized. It's most likely a bacteria. It's most likely a virus. Start making your antiviral or your antibacterial war plans now. And we do. Okay? Okay, I want everybody to stand up and take a break. Talk to your neighbor for 2 minutes. Because you guys are not with me. And this is important. 2 minutes. I'm also going to give you your poll everywhere question. The question is which cell is a phagocytic granulocyte? A Kupffer cell, a mononuclear dendritic cell, a mast cell, an eosinophil, or a PMN? Are you guys all, has it been a long day? Was there a big game I didn't know about? No? It's just, yeah. Everybody is just not with me. This is a question that has been on past exams. Hint. 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 Everybody breathe deep. Are we good? Are we back with me? Okay. I'm going to do this. Are we done? People are still working. I gave you 2 minutes. Okay, 5, 4, 3, 2, 1. Okay, what's the correct answer? Ooh. Okay, let's break down this answer. Okay, the correct answer is a PMN. One, when we go through the answer, let's just break down that question. It's asking for a granulocyte. Is a Kupffer cell a granulocyte? It's a macrophage. Is a mononuclear dendritic cell a granulocyte? It's a mononuclear phagocyte. Is a mast cell a granulocyte? Yes. Is it phagocytic? No. Okay? Is an eosinophil a granulocyte? Yes. Is it phagocytic? No. Is a PMN phagocytic? Yes. Is it a granulocyte? Yes. This is the answer you need to choose. Yes. For a possible exam question. Hint, 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 hint. Okay? Are we good? Okay. So, you guys, I know people say, oh, she expects us to know everything. Yeah, I do. I'm sorry. But what you have to understand is your everything is literally this much of microbiology. We are skimming the surface of so much more stuff that's so much more complicated that we as microbiologists keep saying, so those are the only 6 cytokines you're asking them to know? I'm saying yes, because that's what the book talks about. There's so much more. It's so much more complicated. This is the least acceptable unit of microbiology that I, as a microbiologist, would be allowed to go through my department and talk about it. Okay? So, what else do we need to know about this? Once an RLR is bound, one of the primary things it induces is something called interferon alpha and beta. Interferon alpha and beta is the most important antiviral innate response that we have. Because remember, in one generation time, how many new virions can we make in one generation time? Is it exponential? Is it more than exponential? It's more than exponential. We have to find a way of stopping the virus from overwhelming us while our elite troops are being generated. Because the elite troops for this specific virus have to be generated because these viruses are sneaky and tricky and evil. Evil. Okay? So, interferon is responsible for this mechanism. Interferon, it can be made by any nucleated cell. Any nucleated cell. What's the only cell in the body that can't make interferon? Red blood cells. Only not nucleated one. However, there are specific kind of dendritic cells that can make 1,000 times more interferon alpha and beta than any other cell in the immune response. And do you know what we call those? Interfering producing cells. They can abbreviate those IPCs, right? For interferon producing cells. They can produce 1,000 times more interferon alpha and beta than any other cell. But what it really comes down to is what happens when a cell gets infected by a virus. So what happens when a cell gets infected by the virus binds to the cell, it attaches, and it's going to either endocytose or fuse, depending on what kind of virus it is. Okay? It's going to endocytose or fuse. It's going to get in, and once it gets in, it's going to uncoat in the cytoplasm. And once it uncoats in the cytoplasm, RNA viruses stay in the cytoplasm. DNA viruses generally migrate to the DNA, the nucleus of the cell they're infecting. So, we have these cells. When those viruses start making double-stranded RNAs, and they always do, some viruses are better at inducing a very, very strong interferon response. Some viruses aren't as good at making a very, very strong interferon response. Some viruses actually find ways of trying to inhibit our interferon response, and we'll talk about those in just a minute. So, when that virus makes double-stranded RNA, those RLRs that are in the cytoplasm bind to that double-stranded RNA, it's a pattern. This tells me that there's a virus present in my cytoplasm, and it's really too late for me. I, as a cell, am over. I'm gone. You can have a really good death scene. As the virus slowly overwhelms your body and takes over and sucks out all of your proteins and your DNA to make new viruses to go on and infect somebody else. But because you are you're going to warn your neighbors that you've been infected by a virus. And to protect themselves, get a dog. Okay? Go and get a dog. There's a virus in the neighborhood. Protect yourself. I'm not going to talk about any other kind of weapons here. We're talking dogs. Okay? You're going to go and get a dog. You get a dog, but the dog isn't activated because all you did was get a dog, and it's just a pet. And it's going to live in your cell, and it's going to be fine. So what happens is the cell that's been infected by a virus has sent a signal out to its neighborhood cells, and in some cases it might just be the few cells that live around it, because it doesn't make a lot of interferon alpha and beta. And it says, I've been infected by a virus. Arm yourselves, get some antiviral weapons out there. And they actually bind to a receptor on the surface of a cell. All cells have these receptors. These receptors bind to interferon alpha or interferon beta, and we call them interferon alpha receptors or interferon beta receptors. Once they bind to interferon alpha or interferon beta, they send a signal into the cell. The interferon itself does nothing but warn the cell that there's a virus present. It just warns the neighboring cell. It's up to the cell to arm itself. And that signal is sent into the nucleus of the cell that says, holy cow, there's a virus in my neighborhood. I need to make antiviral weapons. but you're not been infected by a virus yet. So what they're going to do is they're going to make inactive antiviral proteins. You're going to need to know that AVP stands for antiviral proteins, and you're going to need to know that IAVP are inactive antiviral proteins. And it's right here. Here you can see in this picture, we have made inactive antiviral proteins because we have been warned by our neighboring cell that it has been infected by a virus. This first cell, the first cell that's infected by a virus, can't stop it. It hadn't been warned by anybody that there's a virus in the neighborhood. So it is going to make new viruses and it is going to die. And these new viruses are going to be released from this cell, and then they're going to go on to the neighboring cells and try to infect the neighboring cells. These inactive antiviral proteins don't prevent the virus from getting in. They don't prevent the virus from uncoding, but what they do are prevent the virus from making new RNA and new proteins. So they have these inactive antiviral proteins activate to active antiviral proteins, and they destroy viral RNA and viral protein synthesis. And it eventually kills the cell that has activated antiviral proteins in it. And people say, well, why don't we just make interferon alpha and beta all the time? And that way we wouldn't have viruses ever. Has anyone in here ever had the actual flu? Not a 3-day thing. The 8-day, you're on your bed, your hair hurts, your teeth hurts, you ache all over. That is actually not your body on the flu. That is your body having an overwhelming interferon alpha and beta response that says you have been infected by a potentially lethal virus. I need you to shut the heck down, lay on your back, stop mucking about, and fight this infection off because this could kill you. Interferon is they actually use interferon therapy for some long-term chronic infections like hepatitis B. One of the drugs that they use as an anti-hepatitis B drug is interferon beta. So many people who are on hepatitis B refuse to take the drug because it makes them feel so bad all of their life. This is why we don't use interferon alpha and beta. And the other thing is we don't want to utilize that energy to make something that we don't need until we get a viral infection. And it might need that we just make a local interferon response. But with an influenza infection, that is going to send that is a lethal. It sends all sorts of alarm systems out. All of those NLRs send out an alarm system that says, this is a hugely lethal infection. Those interferon-producing dendritic cells start pumping out thousands of molecules of interferon to warn the entire body that any part of this body could be infected and you need to protect yourself. And we do. And then, slowly but surely, you get over the infection. And once you get over the infection, your interferon response, if they weren't activated, those inactive antiviral proteins get degraded and shut away. So it's not a permanent response. It has no basis on the kind of virus it is. It has nothing to do with the specific virus. It doesn't know that it's a measles virus. It doesn't know that it's rubella. It doesn't know that it's the flu. It knows that it's a virus because there's a pattern it recognized, double-stranded RNA, that tells me that this is a virus. Okay? This is the most important immune response against viruses. Viruses fight back. Some viruses make fake interferon alpha and beta receptors that they send out into the environment to soak up the interferon alpha and beta that's being made by a cell that's dying. So it's not as much interferon alpha. How did they know how to do that? It's a virus. It's just genetic material in a protein code. How do they know how to do that? Somehow they learned, and they're using it against us to try and stop our interferon response. Okay? So this is our antiviral response. When we talk about interferon gamma, it's an unfortunate name for a cytokine. It has nothing to do with viruses. It is the cytokine that we make to activate macrophages. When we activate macrophages, they go from the garbage man that's cleaning up dead and dying cells or cell debris to become, and you can kind of think of it as becoming an Incredible Hulk. So you go from normal everyday Bruce Bannon to an Incredible Hulk that is really angry and killy. It wants to destroy something. It is the Hulk. It doesn't really know what it's doing. It's going to try and destroy things. It becomes, and this is an unfortunate drawing because you have to look at the size. This is 2 micrometers, and this is 10. This becomes bigger. It becomes full of toxic molecules. These toxic molecules are reactive oxygen species. We throw a lot of reactive oxygen species in there because these angry, killy macrophages are going to want to eat up bacteria, throw reactive oxygen species, destroy their DNA, destroy their lipids, destroy their proteins, and kill these organisms. Interferon gamma is the cytokine that activates macrophages. It can activate dendritic cells too, but it's activating macrophages that's important. This is the cytokine. Unless you have this cytokine, unless you can make this cytokine, you generally can't fight off something called tuberculosis. They actually do an interferon gamma release response in people who are having outbreaks of tuberculosis to see if they're making normal amounts of interferon gamma because it's macrophages that have to fight off this organism. Are we good? Okay. Here we have activated macrophages. Here we have a normal macrophage. Now, the last thing that we're going to talk about, and we're going to continue about this in the next class. In the next class, we're going to get into the acquired immune response. So make sure you bring the acquired immune response outline. Watch the animations for inflammation. I will ask you a pull everywhere question about that animation for inflammation and complement next class. Okay? So when we talk about complement is an antibacterial response. You can think of these are, I like to think of these as like landmines in your blood. They're floating landmines in your blood. And normally, these landmines in your blood, they're called zymogens. They're inactive enzymes. And as long as they're inactive, they're a zymogen. But if they bump into or if they're activated by a bacterium, they become activated and they form this cascade that is antibacterial specifically. We have found that they can work on some viruses and they can work on more things than this, but in this class and in this particular environment, we're going to say that they're primarily antibacterial. Okay? I want you to know that there are 3 different ways of initiating this pathway. One way is by the bacterium themselves. One way is if a bacterium has something called mannose on its surface. And one way is if a bacterium is coated in antibodies. They make something called convertases, and those convertases act other molecules in the cascade. And we'll go over these on Thursday. Watch the animations. They're good. They don't always make sense. We'll talk more about them. Read your outline. Watch the animations. We'll talk more about them on Thursday. I need to talk to Kim, the people who have appointments with me this afternoon. Melissa, Cayenne, and Michaela. Melissa, Cayenne, and Michaela, come this way.